Did you know that only about 20% of patients worldwide can access the biotherapeutics they need? Let that sink in for a moment. In an age of breakthrough therapies and medical innovations, 4 out of 5 patients who need these treatments can't access them. Welcome to the Smart Biotech Scientist podcast. I'm your host, David Brühlmann, and this statistic isn't just a number to me. It's why I do what I do. After 15 plus years at the forefront of bioprocess innovation, I have seen firsthand how manufacturing costs can put life-saving therapies out of reach for most patients. This reality shapes my mission, helping biotech leaders reduce manufacturing costs while accelerating development and minimizing risks. Because every cost reduction we achieve means more patients can access the treatments they desperately need. Today marks the beginning of a special 4-part series based on my recent keynote at the Amphacademy. We'll explore not just the evolution and the future of bioprocessing, but more importantly, how we can make these breakthrough therapies accessible to more people worldwide. In part 1, we will trace the journey from the early days of recombinant insulin to today's revolutionary therapies. Whether you are working in cell therapy, biologics, or cultivated meat, understanding this evolution is crucial for what comes next. Because we're not just developing processes, we are shaping the future of healthcare and sustainable food production. Let's dive in and explore how you can transform the future of bioprocessing to make these life-changing therapies accessible to all who need them.
I'd like to welcome Dr. David Brühlmann, Unlocking the Secrets of Next-Generation Mammalian Bioprocessing. Now, David is the managing director of his own company, Brühlmann Consulting, and you have a focus on biology and technology and help companies and research institutes to develop that into commercial products.
Thank you very much for this amazing introduction. Good morning. How are you doing? Good to see you all. Let's imagine that we are on a Thursday morning at exactly 7:43 on a Thursday morning in 2030. In a Singapore lab, a technician is just finalizing a personalized cell therapy that began manufacturing just 9 days ago using the patient's own cells. By evening, it will be in Mumbai, ready to save a child's life. At that exact moment, in a Chicago restaurant kitchen, a chef is unwrapping what looks like the perfect Wagyu steak. Some people are getting hungry already. Except this cut never required one single animal. It was produced in a precision-controlled bioreactor just down the street. And in a Berlin hospital, an oncologist is reviewing real-time manufacturing data of a bispecific antibody that's specifically produced for her patient. A scenario that would have been dismissed as scientifically impossible just a decade ago. Ladies and gentlemen, all of these 3 scenarios are already in the making. The biology has been proven, the scientific foundations exist. The one single challenge that is standing between these breakthrough technologies and widespread adoption is manufacturing at scale. And that's why we're here for. Because the next revolution in the bioprocessing industry won't just happen in research facilities. It will happen in our bioprocessing facilities. And it's happening faster than you think. So let me put this into perspective. When the first recombinant insulin was approved in 1982, it was considered a miracle. In the decades to follow, antibody therapeutics have revolutionized treatment options in a vast variety of diseases. And today, We have more than 100 FDA-approved antibody therapeutics treating everything from cancer to autoimmune diseases. But here is where it gets interesting. That revolution was just a warm-up act, because the 2024 biotech industry isn't just about antibodies anymore. It's expanding in directions we would have just simply thought that they were impossible just a few years ago. And they would have probably seemed like science fiction in those early recombinant protein days. Let's pause here for a moment. What we have just covered, the journey from recombinant insulin to antibody therapeutics, laid the foundation for today's innovations. But what's truly fascinating is how this foundation is being completely reimagined for new modalities. As we continue, pay attention to how these early achievements are enabling today's breakthroughs. We are now seeing bispecific antibodies that target multiple disease pathways simultaneously. We have fusion molecules, we have nanobodies, we have DNA and RNA, we cultivate viruses, and we have cell therapies that turn our immune systems into precision cancer-fighting machines. And we have cultivated meat and fish that could revolutionize our food systems. So I would like to hear from you, which of these modalities do you think are the most promising in the future? Is it bispecifics? Anybody that believes it's bispecifics that revolutionize next-gen manufacturing? Not so sure yet. Is it maybe fusion molecules? Not so sure. Is it DNA and RNA? I see some hands. Good. Perhaps cell therapies? Oh, yes, a lot of hands. Okay. At least we agree on that. What we now are seeing is that new modalities outpace the conventional ones in revenue. Here we are looking at the forward projected pipeline value. And if we look at the 2019 pipeline, the 2024 projected value still mainly comes from traditional modalities, about 59%. Now, fast forward that to 2023, to the pipeline, the projected value, the 5-year projected value, will mainly come from new modalities, 56%. When I was working at Merck, we put a lot of effort into enhancing the antibody production. And if you're familiar with antibody production, we have a certain number of seed train reactors in a batch mode to ramp up the volume. And then once you have enough cell, you seed your production bioreactor. And that's typically run in a fat batch mode. Instead of doing the last seed train called N-1 in a batch mode, we switched to a perfusion mode reaction. And this has several advantages. And one of them is that you can extend the culture and you can reach much higher cell densities. And that's great, because if we can reach more cells in the N-1 stage, we can start the production stage with a lot more cells. And inspired by this work, Ichnos, they developed a intensified process to produce a difficult to express multispecific antibody. And they first started by obviously optimizing the cell line itself. And once they had a suitable cell line, they went into the fed batch process first. And like a typical fed batch process, you reach about 20 million viable cells per mil. And they reached about a titer of 6 grams per liter for this difficult to express protein, which is already very good. Now, moving into a high density process, they were able to reach 70 million viable cells per mil. And because of that, they were able to reach titers of about 10 grams per liter. And now for clone C, they also tested the very same process where they were able to reach even a higher titer of 11 grams per liter. On the downstream side, we are facing yet another challenge, because we are moving away from traditional antibody formats. We are now seeing proteins that lack the Fc part of the molecule. And if you know something about proteinase, specifically targets the Fc part of the molecule. And Cytiva recognized this challenge, and they developed a new affinity chromatography resin that also enables high purities, but now for molecules lacking the Fc part. The way they do that is when you develop your protein of interest, the sequence, you add an additional amino acid sequence to it, and then a tag is also expressed at the end of the protein on one of the terminus of the protein. Now, when you load this protein of interest onto your column, again with a traditional antibody, the ligand specifically recognizes the protein, it specifically binds to it, and now comes the genius part. When you elute, not only is the protein of interest released, the tag actually stays on the ligand. So only the protein of interest is released.
And that wraps up part 1 of our 4-part series on the evolution and future of bioprocessing. The transformation we have witnessed from basic recombinant proteins to complex therapeutics is just the beginning. In part 2, we'll dive deep into the 5 critical challenges facing our industry today. Do you want to make revolutionary therapies accessible to more patients? As your strategic advisor with over 15 years of bioprocess innovation experience, I help leaders accelerating and de-risking development and thus reduce manufacturing costs. My expertise enables you to transform complex science into a commercial success, navigate regulatory challenges with confidence, and scale the production while maintaining quality. Book your free 30-minute assessment at BrühlmannConsulting.com and discover how you can accelerate your path to market. Thank you for tuning in. Join us next time as we explore the specific challenges that are reshaping our industry. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.